Today we will learn about another signaling pathway which is also very important and oncogenic also. It's called Wnt or beta catenin signaling pathway. This pathway is especially important as it is important for the development of colon cancer. It contributes to the pathology as well as progression of colon cancer. So, it is very important in that. Of course, it is also important in some other cancers as we will see it as we go along. The Wnt ligands are many as usual, the family of Wnt ligands are known. They generally encode secreted lipoproteins. They act as ligands for a receptor called frizzled receptor FZ. This is also important in development. Wnt is, in fact, the name Wnt came from studies during development. There are development biologists, so they study the development processes in various organisms, and it is coming from that flies wingless is a gene, So from that only they have derived this. So it is important for development of the morphology, morphogenic signaling. What is interesting to us is that some components in this Wnt signaling pathway are either inactivated or mutated. The important components are Wnt's, the Wnt ligands, APC. APC is a tumor suppressor gene, adenomatous polyposis coli gene, then axin and beta catenin. However, the central player is beta catenin. Why? We will see that. This is how it happens. The Wnt ligand binds to the frizzled receptor. You can see it is very similar to that transmembrane serpentine receptors. So, it is called a frizzled receptor. Then it binds to that, and then the signal is passed on to what is called disheveled. Yeah, the Wnt ligands bind to the frizzled receptor, and that signal is passed on to what is called disheveled. Once these are activated, the signal goes to a complex often called death complex. It consists of the idea why we are calling that as degradation complex or death complex is because this is the complex that eventually degrades beta catenin. That is why we call that way. Beta catenin is the main effector molecule that translocates to the nucleus and helps in transcription. So, the components of this complex are axin, the glycogen synthase kinase GSK3, APC. APC I told you is a tumor suppressor. So, this is the complex. When GSK3 beta gets activated ultimately, and this complex sends a signal, basically it phosphorylates beta catenin, and that is a signal for its degradation. We will see that in the next slide. You can see that. See here, without Wnt activation, when there is a no ligand, this complex that I told you, GSK3 beta, axin, APC together, they phosphorylate. Of course, the main the kinase that phosphorylates is GSK3 beta. Okay, that is the kinase, glycogen synthase kinase. It phosphorylates beta catenin at specific N-terminal residues, serine residues. So, it phosphorylates that, and this phosphorylated beta catenin is destined for degradation. So, it will be taken to the proteasome complex where it gets degraded. Now, there is no beta catenin, no Wnt signaling. because there is no ligand, see here. Now, with Wnt activation, what happens? Wnt binds to frizzled and then disheveled. This complex that I told you, GSK3 beta complex, is actually inhibited by disheveled. So, now beta catenin is free to move to the nucleus, bind a certain sequence. Beta catenin needs other factors, coactivators and all that, to bind to specific sequence in the DNA. It does not bind on its own, but it needs the transcriptional complexes. Together they bind DNA, and that results in transcription, which leads to proliferation, because ultimately the transcripted genes include some of the oncogenes like MYC, cyclin D, cyclin D1, P300 and such genes. So, that results in proliferation. So, this is what happens. This has to be understood, it is a very important signaling. So, Wnt signaling basically prevents degradation of beta catenin, and thereby it helps beta catenin to translocate to the nucleus, and then the signaling takes place. So, normally it is kept degraded. It is like it is almost like P53, if you remember. P53 and MDM2 complexes are there. MDM2 keeps it for degradation. So, normally P53 level is kept low. Similarly, here beta catenin level is kept low. When there is a ligand only, you get the translocation of beta catenin, and then, so these are simple mechanisms. Another interesting thing is, many transcription factors act like this. This is one mechanism that you have to keep in mind. See, for a protein to be synthesized and then get activated upon some signal, it is going to take a lot of time. So, one easy way the organisms have devised is to keep those transcription factors in some inactive state. So, whenever you need their function, you activate them by removing that inhibition, so that they are immediately available for transcription. They do not have to be synthesized. So, this is one mechanism. Later on, I will tell about NF kappa B, other transcription factors and so on. By the way, beta catenin is not directly a transcription factor. As I told you, it needs other proteins to bind to the DNA sequence. When you say transcription factor, it is going to bind a specific sequence. Here it is not like that, it needs other factors. So, that has to be kept in mind. But whenever you need transcription, this is one important mechanism by which many transcription factors act. That is what I am trying to say. Do not confuse it with just beta catenin as a transcription factor or something like that. It is not. Now, this is a APC mutation or beta catenin mutation is very common in colon cancers, as I told you. So, what happens is many of the cells, they get arrested, and then slowly they become undifferentiated progenitors. So, these changes are necessary. If you remember, I told you several changes occur before a normal colon cell becomes a colon cancer cell. There are steps involved, there are mutations. Under that, I have told you about mutation of APC also. This is almost in all the patients you see APC mutation. This is a very common thing, but then you have to remember, it is not that this mutation is responsible for cancer. No, you need additional changes. So, they almost follow the Knudsen's hypothesis that some of the familial cancers, you have one allele mutated and then another allele also has to be mutated to get a full-fledged cancer. That happens in some of the familial cases. Here, of course, APC mutation is one triggering event for other changes to happen, and stem like cells are formed. See, this is the colon structure they have given. What happens is there are polyps that are formed. Polyps, if you remember, I told you they are like small protrusions in the colon. So, that is the first step. That is why it is called adenomatous polyposis coli, we say. So, APC is responsible for that. So, those small projections are formed initially, and then they develop into more serious condition, and finally the uh, uh, cancer develops. Similarly, some stem cells also play a role in that. So, you see the uh, interaction between different cells in this, for example, stem cells, some of the stromal cells, and the colon cells, these these uh, protrusions that I told you just now. So, those are necessary ultimately to develop a full-fledged cancer. Overexpression and activation of growth factor receptors like EGFR results in the activation of the MAP kinase pathway. We have seen that. Similarly, mutation of RAF proteins can also result in constitutive MAP kinase signaling. This is slightly in between these sentences are coming, but just do not worry. Expression of WNTs increased 4 to 10 fold in many breast cancers. Increased nuclear translocation of beta catenin is seen in more than 20% of the prostate cancers, not only in colon cancers, but also in other cancers, as I told you. Some prostate cancers have a mutant beta catenin that is resistant to phosphorylation by GSK3 beta, increasing its half life. Because you see that phosphorylation by phosphorylation of beta catenin by GSK3 beta is necessary for its degradation, right. So, that if that is prevented, then beta catenin will not get degraded and will move to the nucleus. You have to remember that. So, if it is resistant to phosphorylation, then what will happen? It will be able to move into the nucleus and will become constitutively active. That is the idea. Defective APC protein leads to defective beta catenin degradation. That is a very, in virtually all colon cancers. Just now I told you that in some familial forms, this polyp to colon cancer sequence is happening after the APC gets mutated. One allele of APC gene is lost in familial polyposis, and so the other one, when it is getting mutated, you get a full-fledged cancer. So, as we discussed, it is following the so-called Knutson's two-hit hypothesis. Now, Wnt signaling, I told you, is responsible for the pathogenesis and development of colon cancers. So, many Wnt inhibitors are used as helpful, are helpful in the treatment of colon cancers. People are looking for Wnt inhibitors. Many naturally occurring compounds also act as Wnt signaling inhibitors. Even from our lab, we have published that plumbagene can act as a Wnt signaling inhibitor. So, there are many such chemical inhibitors which are also useful in therapy. It is not that they will be directly useful, they are usually given along with whatever is the standard treatment, chemotherapy or something like that. It is usually for combination therapy, people are looking for Wnt signaling inhibitor. That is an important point for you to note down. So, that is not shown in this slide, so that is why I just said that. Okay. The next pathway, the so-called cytokine signaling and the JAK-STAT pathway, we will look at it. The cytokines, as you know, are very important for immune system. They have an important role in the immune system, neuronal and hematopoietic and embryonal development also. Some of the cytokines include the so-called interleukins, erythropoietin, interferon, and the very important one, tumor necrosis factor. These are some of the important ones. Later on, I will talk about tumor necrosis factor in connection with apoptosis also. It can induce apoptosis. So, that pathway we will discuss afterwards. Often they act locally, meaning that they act on the cells that as the cytokine producing cells. We'll look at some examples so that it will be slightly clear. If you remember, when I talked about receptors, I told you some receptors will have tyrosine kinase activity. They are called receptor tyrosine kinases. But some other receptors, they will not have intrinsic tyrosine kinase activity, but they will be transmembrane proteins, but they will align with intracellular tyrosine kinases. So, ultimately they function almost like receptor tyrosine kinases. The only difference is receptor tyrosine kinases have intrinsic tyrosine kinase activity. These receptors, immune receptors, they do not have intrinsic tyrosine kinase activity, but they align with cytoplasmic tyrosine kinases. Once this partnership is ensured, they are almost like receptor tyrosine kinases. Do you follow this? This is a very important concept to understand. So, they have no intrinsic kinase activity. Instead, ligand binding leads to conformational change in the cytoplasmic domain of the receptor and activation of the tyrosine kinase that is associated with the receptor. So, these conformational changes when the ligand binds to the receptor leads to attracting the cytoplasmic tyrosine kinase and getting it activated. So, that is how that tyrosine kinase becomes active, and that ultimately phosphorylates. One such tyrosine kinase is called Janus kinase. There are many kinases, and people have called this as Janus kinase. Janus because it has, Janus is a what? Have you heard of that? Greek in mythology, it contains two phases, two different phases. One, I don't remember what is the what are the two different phases, but it has got two phases, the god. Just like our Hindu gods also, you know, like Ganapati having an elephant head and human body, something like that. So, it is similar to that. That is because here it is attaching with two different things. One is the receptor and the other molecules that are interacting. So, they call it. Of course, initially when many kinases were known, this was also called just another kinase. That is why it is JAK, JAK. That is anyway, you can read that in some how these kinases were discovered and all that. If you go through that, you will see such interesting stories. So, JAK, it is a family of proteins or such tyrosine kinases that are associated with. JAK is an important tyrosine kinase. It is associated with immune signaling, immune receptors, cytokine receptors. So, JAKs can phosphorylate certain transcription factors. Again, there are many such transcription factors, STAT1, STAT3 and so on. So, they are called STATs. STAT means signal transducers and activators of transcription. That is the full form for STAT. Signal transducers and activators of transcription. That is what is called STAT. There are many STATs, STAT 1, 2, 3, 5, something like that. So, JAKs can phosphorylate STAT, and this in addition to other substrates in other mitogenic pathways. So, this is an important point. Now, we will look at the STAT proteins. As I told you, STATs are transcription factors basically. They are found in the cytosol. They can be activated by the tyrosine kinases such as JAKs or even by receptor tyrosine kinases and also by non-receptor tyrosine kinases such as SRC. JAK is also a non-receptor tyrosine kinase because it is not a receptor. SRC is also not a receptor, but a tyrosine kinase. Do not think whenever I say tyrosine kinase, this is a confusion students have. They think they are all receptors. No, that is why we distinguish some tyrosine kinases as receptor tyrosine kinases and non-receptor tyrosine kinases. Non-receptor tyrosine kinases are usually present in the cytosol, whereas receptor tyrosine kinases are present in the membrane. They are transmembrane protein. Receptor tyrosine kinases typically have an extracellular portion, transmembrane portion, cytoplasmic portion that has a tyrosine kinase activity and a tail, cytoplasmic tail that contains tyrosine residues. So, these are important points that you have to kind of remember. And they can be activated by GPCRs also indirectly. So, that means they are one component of the signaling pathway of GPCRs, that is what STAT. So, STATs are very important. They can be phosphorylated by JAKs, receptor tyrosine kinases, non-receptor tyrosine kinases such as SRC and GPCRs. Now, what the STATs do? STATs activate transcription of MYC, cyclin D2 and D3 and BCL-XL. BCL-XL by the way is an anti-apoptotic gene. You have heard of BCL2, it is an anti-apoptotic factor. Later on we will study that in detail, but BCL-XL is also another anti-apoptotic gene. So, that those are, see, anti-apoptotic gene is also one way contributes to proliferation. I told you. What is an anti-apoptotic gene? It increases the survival. If it increases the survival of the cancer cells, indirectly you are helping cell proliferation, because it is the cells that are having this are already cancer cells and you are making it to live long. That means you are indirectly promoting cell proliferation. So, many of the anti-apoptotic genes are even considered as oncogenes because of this property. They are not actually oncogenes, they are survival genes rather. They can be termed as survival genes, but then because they promote the survival of cancer cells, they are also often considered as oncogenes, such as MDM2 for example. MDM2, it is anti-apoptotic because it degrades P53, which is pro-apoptotic. So, that way when MDM2 is overexpressed in certain cancers, they also act as oncogenes in that sense of promoting the survival. So, that has to be kept in mind. JAK-STAT pathway is slightly different from other pathways such as the RAS MAP kinase pathway. If you remember, I told so many intermediates, GRAP2, SOS, RAF, MAP kinases, then that ribosomal S6 kinase and finally MYC and other genes. So, it contains so many intermediates, whereas in the cytokine signaling nothing, just the receptor and then immediately JAK STAT and then the transcription. So, only very few members. Is there any advantage of having small number of components and instead of having a lengthy list of signaling intermediates? Yes, because short pathway you end up your duty very quickly, correct. If there is a function it will not take long time, because if there are long intermediates, it's going to take some more time to do whatever function the pathway is intending to do. So, that way it has advantages. The disadvantage is perhaps the amplification of signaling, diversification. Those are possible. If you have many intermediates, you will be able to amplify the signal, you can make it a big circuit and so on. So, those are some of the points that you may have to ponder over. There are other such kinases called type 2, JAK. So, this is one typical thing. STATs are found in latent form in the cytosol, which means they are inactive form. They are activated by tyrosine kinases such as JAK, this type 2. Type 2 is also a kinase, tyrosine kinase. So, they can phosphorylate. Like I told you, these transcription factors are multiple forms are there. STAT1 and STAT2, they form a dimer. For activation, you need this dimeric form. It is this dimeric form that translocates to the nucleus. So, activated by tyrosine kinase, phosphorylation at around 700 residues from N terminus. That means, the protein is a big protein, more than 700 amino acids are there. So, around that is in the tail portion you have the tyrosine phosphorylation, that is what, just like any other receptors also. In EGF receptor it may be even 1000 residues, so it is not surprising. So, activation leads to STAT dimerization. This is the one that enters the nucleus and activates transcription. So, that is an important. Now, some of the aberrations in cancers, in many cancers, STAT3 is constitutively activated. Melanoma, for example, there is a constitutive activation of SRC. Majority of breast cancers, STAT3 is constitutively active. Head and neck cancers, people have tried to do certain site directed mutagenesis of 2 STAT subunits, so that if you attach just this is to prove the point that dimeric STAT is very active. So, people have artificially attached 2 STAT subunits covalently, and that acted as a constitutively active dimer. So, that is what is shown as the so called butterfly structure, and that gets activated. STAT3 dimers are activated. Okay. We are unless you have some questions, we will kind of stop here, because I have to go also. Unless you have some questions, you can ask. We will stop here today. Today is a little bit short, but we will take up other signaling pathways later.